exist other pathway synthesis of ammonia in the human body, not only deamination of amino acid. The most commonly among other pathway production of ammonia, there is processes deamination of nitrogen bases of mononucleotides, processes deamination of biogenic amines in the tissue. And please note what certain role in the providing ammonia in the human body take part bacteria in the large intestine, which produce ammonia, and that ammonia may be under absorption too. So it's about sources of ammonia. Now, my dear colleagues, about other very important problem, about toxicity of ammonia. Ammonia is very toxic substances. My dear colleagues, please note on the reference level of ammonia in the blood, the normal level of ammonia in the blood of adult healthy person. As you can see, it's very low level in micromolar range concentration. Please note, the most of metabolites in the human body contained in the blood in millimolar concentration, about 1,000 times more. So it's toxic substances. For that reason, its normal concentration is very low, And then concentration of ammonia in the blood increase more than 32 micromole per liter. Appear clinical manifestation intoxication of ammonia. And my dear colleagues, please note, the most sensitive to intoxication of ammonia, there is central nervous system. As a result, intoxication of ammonia appear such symptoms like preferentially it's maybe it's maybe convulsion, it's maybe excitation, it's maybe comatose state, and often intoxication of high level of ammonia bring to death of such person. My dear colleagues, first of all, let's say about mechanism toxic effect ammonia in the central nervous system. Excuse me, I give permission work in lecture with other student. So let's carry on. You have, I hope you know, such as previous session, you pass through exam on biochemistry and, through exam on histology and anatomy. And you know what in the brain exists two kinds of cell. First of all, it's neurons, and neurons provide appear high-level function of the brain. And glial cell. Among glial cell exists special kind of such cell, such astrocytes. Astrocytes is very important in the providing neuron with building blocks for synthesis own molecule in neuron. And the astrocyte accept products of catabolism from neuron and later pass them to the blood, and via bloodstream, these products, catabolism of neuron, later eliminate from human body, for example, with urine or with bile. Exists different way elimination. So Why I say you about different kind of cell in the nerve system, central nervous system? You must know the most toxic effect of ammonia coupled with injury astrocytes, glial cell. Let's say about injury metabolic processes in astrocytes during intoxication of ammonia. My dear colleagues, then concentration of ammonia sharply increase. Ammonia must be undergo binding in different metabolic pathway. Exist two most important pathway binding of ammonia. Later we will say in detail about this pathway. But now I want to say you about interaction ammonia with alpha ketoglutarate in reductive amination of alpha ketoglutarate resulted from that ammonia binds with alpha ketoglutarate resulted from a rise in toxic substances, glutamate. Glutamate is very important substances, nitrogen-containing substances in the nerve cell and take a pivotal role among metabolism of other amino acid too. Metabolism of most amino acid in the nerve cell and glial cell coupled with metabolism of glutamate. But then concentration of ammonia sharply increase, and that processes will be not enough for complete binding of ammonia. Ammonia later will bind with glutamate. Resulted reaction amidation of glutamate. Arise glutamine, ammonia bind in the side chain of glutamate resulted from arise glutamine. Please note that process is energy-dependent process and require ATP. Resulted from arise glutamine. My dear colleagues, glutamine is a non-essential amino acid like glutamate. It's in toxic substances. For any cell and for cell of central nervous system too. But then inside brain synthesis cells of brain synthesis a lot of glutamine. It may be then concentration ammonia during intoxication is very high. Concentration glutamine sharply increase, and accumulation of glutamine bring to increase osmotic pressure inside cell, inside astrocyte, for example, resulted from increasing of osmotic pressure bring to trigger water inside cell, resulted from arise cerebral edema. And cerebral edema is very dangerous, such as brain restricted in the cave of skull, resulted from increased intracranial pressure. And increasing intracranial pressure and appear edema of brain bring to arise comatose state, unconscious state. So it's very dangerous condition, and only appear that changes maybe bring to death of patient which undergo intoxication of ammonia. But it's not only one way intoxication, injury, nerve cell during intoxication of ammonia. You must know what increased concentration of ammonia bring to appear disorder of neurotransmitter system. Why is it? My dear colleagues, I said you what intoxication of ammonia bring to decrease concentration of glutamate, such as glutamate transform into glutamine. Glutamate uses as one of neurotransmitter in the central nerve system. From other side, glutamate, what concentration is decreased during intoxication of ammonia in the brain via decarboxylation transform into gamma aminobutyrate. And my dear colleagues, I hope you know what gamma aminobutyrate is very important break neurotransmitter in central nerve system. For that reason, I want to say once more, accumulation of ammonia, intoxication of ammonia, bring to sharply injury of neurotransmitter function of neurotransmitter system, resulted from increased process excitation in the brain, and again appear condition for arise convulsion. But it's not all according to intoxication. My dear, intoxication of ammonia. During intoxication of ammonia is very important decrease energy supplement of nerve cell. Why is it? My dear colleagues, please note, as I said you before, reductive amination, what uses for binding of ammonia with alpha ketoglutarate, bring to decrease concentration of alpha ketoglutarate in nerve cell, resulted from decrease process oxidative transformation intermediating citric acid cycle. And citric acid cycle, as you know, is one of most important process, metabolic process in the energy supplement of cell. From other side, process binding of ammonia coupled with using ATP in reaction, for example, synthesis of glutamine from glutamate. But simultaneously, processes coupled with biosynthesis ATP inside nerve cell decrease. Why is it? First of all, it's alkalosis. My dear colleagues, please note, alkalosis appear during intoxication of ammonia. As a result, increased concentration ammonia in the blood, in the cytoplasm of cell. In the blood, it's bring to decrease providing nerve cells with oxygen, such as alkalosis, bring to increase affinity hemoglobin to oxygen, resulted from oxyhemoglobin don't undergo process dissociation to yield free oxygen, resulted from arise hypoxia, decrease level of oxygen inside nerve cell. As a result of hypoxia, decrease function of mitochondrial respiratory chain in nerve cell, in mitochondria of nerve cell, resulted from decreased rate of process oxidative phosphorylation. You know what process oxidative phosphorylation is the most important process in the providing cell with ATP. So now I want some, my dear colleagues, then concentration of ammonia sharply increase. It's bring to accumulation of glutamine in nerve cell resulted from a rise cerebral edema and intracranial pressure increase, a rise comatose condition. From other side, simultaneously appear disorder in neurotransmitter metabolism resulted from, again, appear condition which bring to excitation and appear of convulsion. And in third, intoxication of ammonia accompanied by decrease of energy supplement of nerve cell. And you must know what nerve cell require a high amount of energy in order to provide their function coupled with generation nerve impulse and their carrier cross axons. So, my dear colleagues, it's about intoxication of ammonia. Modern view about intoxication of ammonia. Next question about detoxification. My dear colleagues, I want to say you once more, what ammonia is normal product catabolism of amino-containing organic substances. For that reason, during process evolution, was created special mechanism detoxification that very toxic product inside cells of human body and inside human body. And you must know what exists different way detoxification of ammonia. Please note, exists next pathway ammonia detoxification, reductive amination of alpha-ketoglutarate, synthesis of amino acid, urea cycle in the liver, and formation of ammonia salts. My dear colleagues, please note, we said already about mechanism detoxification of ammonia by reductive amination of alpha-ketoglutarate. We said about mechanism amidation of amino acid. It may be synthesis of glutamine and asparagine. And please note that processes may effectively bind ammonia only then concentration of ammonia will be very low, such as in that condition, that mechanism will be enough for binding of ammonia. But then concentration of ammonia sharply increase, effectivity of that process in detoxification of ammonia won't be enough. For that reason, exists also one specific pathway detoxification, which called urea cycle. Urea cycle. My dear colleagues, please note, urea cycle is tissue-specific process. It takes place inside liver. For that reason, ammonia, which synthesizes during processes deamination in different tissue of human body, might be transferred from the tissue inside liver. But in order to provide that transport, needs specific pathway coupled with transformation ammonia in toxic substances which uses for carrier of the ammonia in the liver. Next question about transport ammonia from tissue in the liver. As transporter of ammonia maybe uses glutamine, asparagine, and alanine. My dear colleagues, again, I show you the same slide what I said have showed you earlier. See here, ammonia, for example, it's maybe in nerve cell, bind with glutamine, transform in glutamate, transform into glutamine. Glutamine may cross cell membrane in certain amount and pass into the bloodstream. Glutamine is not toxic substances. It's some substance. It's moreover non-essential amino acids, which may be used as cell for synthesis of protein and other processes. Glutamine leave visceral organs or nerve system, pass into the bloodstream, accepted by hepatocytes. Here it's undergo process hydrolysis in reaction catalyzed by enzyme glutaminase. It's enzyme which catalyzes hydrolytic process breakdown of glutamine to yield glutamate and free ammonia. But that free ammonia appear in the liver, where take place the most important process, detoxification, urea cycle. Exists other transporter of ammonia. I said you about asparagine in the same manner. I don't say you about that. And alanine may be used as transporter of ammonia too. My dear colleagues, how alanine may be used for that purpose? See here, free ammonia in the tissue of visceral organ, for example, in muscle tissue, bind with alpha-ketoglutarate in reaction reductive amination, resulted from a rise in toxic glutamate. Later, glutamate in such tissue like muscle may involve in alanine aminotransferase reaction, resulted from amino growth from glutamate pass to pyruvate, resulted from a rise alanine in reaction catalyzed by that enzyme, alanine aminotransferase. Alanine is in toxic substances too. Alanine leave muscle cell or cell of other visceral organ, passing to the bloodstream and accepted by liver. In liver, alanine again involved in alanine aminotransferase reaction. That reaction, reversible reaction, and in liver cell, they pass in the direction synthesis of glutamate. Amino group of alanine bind with alpha-ketoglutarate, rise glutamate in the liver cell. And later, glutamate involved in glutamate dehydrogenase reaction in liver cell resulted from glutamate undergo process oxidative broken to yield alpha-ketoglutarate and free ammonia, and simultaneously here arise reduced NAD, what can be used as a substrate oxidation for mitochondrial respiratory chain. So that ammonia appear in the hepatocyte, and here it's involved in the process urea cycle. My dear colleagues, between process synthesis of intoxic carrier of ammonia in muscle cell and releasing ammonia in liver, exists specific interaction. That kind of interaction will be designated as glucose-alanine cycle. What's mean glucose-alanine cycle? It's cycle conversation, different metabolite which take place in muscle cell and in liver. See here, in muscle cell, in glycolysis, glucose transform into pyruvate. Pyruvate uses for acception of ammonia transform to alanine in reaction what I said you earlier. Alanine pass into the bloodstream, accepted by liver, absorbed by liver cell, resulted from ammonia release and involved the process coupled with the detoxification in urea cycle. Pyruvate transformed into glucose in the liver cell in gluconeogenesis. Glucose may leave liver cell, pass into the bloodstream, accepted muscle cell, resulted from, again, uses as source of energy and provide muscle contraction. So between that interaction in muscle cell in liver exists appear specific metabolic cycle, and that metabolic cycle designate as glucose-alanine cycle. Glucose-alanine cycle, please note, accomplish two main functions. First of all, that cycle provide transport ammonia from muscle cell into the liver, and from other side, other function of glucose-alanine cycle, coupled with provided muscle cell with glucose, which appear as in the process gluconeogenesis from pyruvate in hepatocyte. So it's about glucose-alanine cycle. My dear colleagues, let's return to process detoxification of ammonia. So in the liver, ammonia, which appeared during process deamination in liver cell, and the ammonia which absorbed from the blood as component of a special transporter of ammonia, involved in the urea cycle. Urea cycle is special process which coupled with transformation of ammonia into intoxic urea. Urea later will undergo elimination from the human body. So, my dear colleagues, let's say about chemistry of process of urea cycle. In first reaction of urea cycle, which catalyzed by carbamyl phosphate synthase, ammonia and carbon dioxide in energy-dependent manner uses for synthesis carbamyl phosphate. My dear colleagues, I hope you know what carbamyl phosphate is macroergic substances. So this bond in the molecule accumulate a lot of energy. And please note, carbamyl phosphate synthase, it's allosteric enzyme, and as allosteric activator of that enzyme, use an acetyl glutamate. Concentration of N-acetyl glutamate appear inside hepatocyte. In condition, then in cytoplasm of hepatocyte appear a lot of free amino acid. So that reaction will be sharply increased, rate of that reaction will be sharply increased in situation then in the cytoplasm of hepatocyte, increase level of free amino acid. Next, carbamyl phosphate involved in reaction catalyzed by ornithine transcarbamylase. In that reaction, uses specific amino acid, ornithine. Ornithine is a non-standard amino acid. They never use for synthesis of protein, but they very important in the liver in the process detoxification of ammonia. As product of that reaction, arise citrulline, other non-standard amino acid. Later, citrulline interact with aspartate in reaction catalyzed by enzyme arginine succinate synthetase. That reaction require energy, and a source of energy uses ATP. Product of that reaction, arginine succinate. Arginine succinate in next reaction, which catalyzed by arginine succinate lyase, undergo process breakdown to yield amino acid arginine and fumarate. My dear colleagues, please note, arginine is a standard amino acid. So arginine may be synthesized in the hepatocyte, but please note what all synthesizes arginine in that process later involve in end reaction of ornithine cycle, resulted from in reaction catalyzed by arginase, arginine undergo hydrolysis resulted from a rise and product of that process, urea, and again appear arnithine. Arnithine may again involve in the ornithine transcarbamylase reaction and uses in that processes. For that reason, that process has cyclic character. For that reason, process synthesis of urea from ammonia, it's so-called urea cycle. In next slide, you can see next net equation of urea cycle. Please note, synthesis only one molecule of urea require a high amount of energy, hydrolysis of 3 molecules of ATP. And also, my dear colleagues, urea cycle tightly binding with Krebs cycle. See interaction between two Z processes. Arginine-succinate-lyase reaction accompanied by release fumarate involved in the citric acid cycle resulted from a rise oxaloacetate, and oxaloacetate in the aspartate aminotransferase reaction bring to synthesis aspartate, and aspartate involved in the process by synthesis of arginine-succinate in urea cycle. So in hepatocyte, the two metabolic processes tightly bind to each other. My dear colleagues, now about role of urea cycle. Then you talk about role of urea cycle. Usually, you know about its role in detoxification of ammonia. It's very important, but it's not only a lone function of that metabolic process. Next function of urea cycle, it's maintain of acid-base balance, such as in urea cycle involve basic and acid products of metabolism. As end product of urea cycle, appear urea. Urea is very interesting metabolite, and the concentration urea in the blood stabilized on the certain so-called reference level. You can see here reference level. And in different conditions and at different diseases, concentration of urea in the blood may be changed and reveal change concentration of urea in the blood may be used in the diagnosis of different diseases. In order to know clinical diagnostic role of urea, you must know what urea synthesizes in the liver from ammonia, and ammonia created from deamination of amino acid preferentially. And urea undergo elimination from human body via kidney with urine. So, if you know that, you must know about clinical diagnostic role of urea. Increasing concentration of urea in the blood may take place at individuals what uses enriched protein diets. So its physiological condition coupled with changed diet. But increased concentration of urine in the blood may be accompanied by different condition diseases, which accompanied by increased tissue proteolysis. For example, it may be during fever, it may be during sepsis, it may be in many other conditions too. Next, renal failure will bring to increase level of urea in the blood, such as renal failure may bring to process decrease filtration of urea from the blood into the urine. And also, increase urea in the blood may be a different diseases and physiological condition which accompanied by dehydration of human body. For example, it may be at burn diseases. It may be then patient don't drink equal amount of water, especially then localizes at hot temperature, and so forth. Next, decrease level of urea in the blood may be again accompanied by disorder in the diet of individuals. For example, low-level protein diet will bring to So, my dear colleagues, let's get started. Next part of my lecture. Now I give you demonstration. I hope you can see my slides. Yes? Can you see my slides?
Yes.
Okay. So, my dear colleagues, about decreasing level urea in the blood and its clinical diagnostic role. I said you about disorder in the diet, using diet with low amount of protein will bring to decrease level urea in the blood. But it may be coupled with different diseases. For example, with diseases of gastrointestinal tract, which accompanied by disorder absorption in the intestine. And diseases coupled with injury of liver, such diseases as hepatitis, such diseases as cirrhosis of liver, such diseases as intoxication of hepatotropic poisons, and so forth. It's about clinical and diagnostic role, quantification urea in the blood. My dear colleagues, now I won't say you what not all ammonia which synthesizes in human body transform into the urine. Some amount of ammonia may be transported in the structure of glutamine, not only in the liver, they may pass into the kidney. And here in the kidney exists special process which designated as ammoniogenesis. My dear colleagues, please note, in the epithelial cell of tubule of nephron, glutamine undergo involved in glutaminase reaction resulted from release one ammonia molecule, and this ammonia molecule later eliminate in the urine. And glutamate in glutamate dehydrogenase reaction here in epithelial cell 2, again transform into alpha-ketoglutarate and release second ammonia molecule. So these two ammonia will be eliminated immediately in the urine and later leave human body with urine. So it's other way elimination of ammonia. The same mechanism elimination coupled with transport in glutamine may be coupled with elimination ammonia via intestine, but we won't say about that process. So next our question will be devoted to special diseases which are called hyperammoniemias. My dear colleagues, what's mean hyperammoniemias? Hyperammoniemias are defined as condition which manifested by increased level ammonia in the blood. So the concentration of ammonia in the blood increase, that condition will be designated as hyperammoniemia. My dear colleagues, exist three kinds of hyperammoniemia: primary, hereditary; secondary, acquired; and transitory. Transitory appears only at newborn child as a result disorder energy supplement of ammonia detoxification in the liver at premature babies only. Let's say about primary and secondary hyperammoniemias. Primary hyperammoniemias appear as a result genetic defect in the genes which encoded enzymes detoxification of ammonia, enzymes which take part in the urea cycle, first of all. My dear colleagues, please note, usually primary hyperammoniemias coupled with sharply decreased production of enzymes as a result disorder expression genes appropriate enzyme. And exist different types of primary hyperammoniemias. Type 1, coupled with appear defect in the synthesis carbamyl phosphate synthase. Type 2, which appear as a result genetic defect ornithine transcarbamylase. Citrullinuria as a result defect synthesis of arginine-succinate synthase. Hyperargininemia, which appear as a result defect production enzyme arginase. My dear colleagues, all that enzymes need for transformation toxic ammonia in toxic urea. For that reason, disorder in the production any enzyme in the in the urea cycle, which bring to a rise primary hyperammoniemias. Primary hyperammoniemias manifested immediately after birth of patient, and clinical manifestation of hyperammoniemia coupled with injury function of brain, arise so-called episodic encephalopathia, and encephalopathia manifested by convulsion and ataxia. So, my dear colleagues, secondary hyperammoniemia arise as acquired diseases. They may appear as a result alteration of liver. For example, hyperammoniemia may be accompanied by such diseases of liver as hepatitis, acute hepatitis, and so forth. So, my dear colleagues, some word about biochemical diagnosis of hyperammoniemias. So, what manifestation in the change biochemical parameter in the blood will be present at patient with hyperammoniaemia. First of all, it's increasing ammonia in the blood, the most specific sign, symptoms, increase ammonia, level ammonia in the blood. In second, it's will be a rise metabolic alkalosis, such as accumulation of ammonia bring to change pH of the blood, And in third, hyperammoniaemia's development as a result, impossible interconversion of ammonia into the urea. For that reason, third specific manifestation of the disease will be decreased concentration of urea in the blood. So I want to summarize. Hyperammoniaemia manifested by increased ammonia in the blood, decrease urea in the blood, and appear or development of metabolic alkalosis. And also about principle of treatment hyperammoniaemia. First of all, it's correction of diet. Such patient must not contain a lot of protein. Diet such patient may contain small amount of protein only. And also administration some drugs which bring to decreased concentration of ammonia. For example, as such drugs may be uses benzoic acid, benzoic acid in the human body, in the liver, bind with glycine, resulted from a rise of hippuric acid, which eliminate from human body. But glycine need for synthesis of protein. For that reason, after binding glycine, need synthesis new molecule of glycine. And synthesis new molecule of glycine require free ammonia. For that reason, ammonia in that condition, a patient with prescribed benzoic acid will be bind ammonia in the processes coupled with biosynthesis of amino acid. As other drug for that purpose may be used glutamate, which may transform into glutamine binding of ammonia, and phenylacetate. Later we will say about phenylacetate, why it can use for binding of ammonia. So it's about hyperammoniaemia. Now, my dear colleagues, next very important part of our lecture will be devoted to metabolism separate amino acid. First of all, we will say about metabolism of phenylalanine. My dear colleagues, what's mean phenylalanine? Phenylalanine, you can see formula of phenylalanine on that slide. It's aromatic amino acid which belong to protein-genic amino acid, standard amino acid, and it's very important to know it's essential amino acid. So phenylalanine cannot be synthesized in the human body. For that reason, protein synthesis provided with phenylalanine in the human body only via exogenous phenylalanine. Exogenous phenylalanine penetrate in the gastrointestinal tract and later undergo absorption in the small intestine. So it's about phenylalanine. Next, about pathway utilization of phenylalanine. What for need phenylalanine? My dear colleagues, then you said about standard amino acid. First of all, you must say about such very important pathway, their utilization, coupled with biosynthesis of proteins and peptides. So phenylalanine, first of all, need for synthesis own proteins in the tissue of human body. All proteins in the human body contain in their structure phenylalanine. For that reason, one of the most important functions of phenylalanine, pathway utilization, coupled with synthesis protein and peptides. Next, phenylalanine may be used for synthesis non-essential amino acid, tyrosine. Tyrosine, later we will say non-essential amino acid, standard and protein-genic amino acid. And synthesis of tyrosine from phenylalanine, coupled with also one very important pathway utilization of phenylalanine. It's catabolism coupled with decomposition to yield end product of catabolism, such as carbon dioxide, ammonia, and water. And why that process is very important, and you must obligatory say about that process. Catabolism of amino acid accompanied by release energy. For that reason, breakdown of amino acid in the human body may be used as very important process in the providing cell with energy too. So, my dear colleagues, now let's say about metabolic processes coupled with transformation of phenylalanine. I said you what process catabolism of phenylalanine and its transformation into tyrosine as first stage process catabolism of phenylalanine. Process catabolism of phenylalanine take place in reaction catalyzed by special enzyme phenylalanine hydroxylase, or other name phenylalanine 4-hydroxylase, or exist also else one synonym phenylalanine 4-monooxygenase. All that synonyms you may use in the same manner. Let's say about transformation phenylalanine in tyrosine. Please note, please see on that slide. As you can see, that process require free oxygen, and enzyme phenylalanine hydroxylase use as coenzymes tetrahydrobiopterin, resulted from effect of that enzyme on phenylalanine. It's undergo oxidation in para position, resulted from a rise tyrosine. One atom from molecule of oxygen uses for formation this hydroxyl group at 4 position, para position. And second atom uses for reduction to water. And that process require coenzyme tetrahydrobiopterin, and oxidation of this coenzyme bring to arise dehydrobiopterin. Dehydrobiopterin cannot involve in that process. For that reason, there is need its reduction to tetrahydrobiopterin again. And exists special enzyme in cytoplasm of cell, dehydrobiopterin reductase, which catalyze process reduction of dehydrobiopterin in tetrahydrobiopterin. And as coenzyme of dehydrobiopterin reductase uses reduced NADP. Reduced NADP uses for reduction dehydrobiopterin into tetrahydrobiopterin. So, resulted from presence that enzyme oxidizes during process transformation phenylalanine into tyrosine dehydrobiopterin, again reduce into tetrahydrobiopterin. So that process, transformation of phenylalanine into tyrosine. And again, please note, this reaction, first stage, catabolism of phenylalanine and tyrosine to end products of catabolism. See here, you must know about that process. Tyrosine later involved in tyrosine transaminase reaction resulted from a rise para-hydroxyphenylpyruvate. Para-hydroxyphenylpyruvate later undergo process oxidation to yield homogentisate or homogentisedic acid. Homogentisate later again undergo process oxidation to yield mallyl acetoacetate. Mallyl acetoacetate transform into formallyl acetoacetate, and after its synthesis, it's undergo process decomposition to yield fumarate and acetoacetate. So, end products catabolism of phenylalanine, there is fumarate and acetoacetate. My dear colleagues, please note, fumarate is intermediate of citric acid cycle. For that reason, fumarate involved in citric acid cycle, and here undergo process degradation to yield end product of catabolism. Acetoacetate later may undergo conjugation with coenzyme A, resulted from a rise acetoacetyl coenzyme A. Acetoacetyl coenzyme A in thiolase reaction undergo broken to yield two molecule acetyl coenzyme A, and both that products decomposition acetoacetyl coenzyme A again involved in citric acid cycle. So all products catabolism of phenylalanine and tyrosine transform into fumarate and acetyl coenzyme A, which involve in the process decomposition in citric acid cycle. For that reason, they may be used as a source, very important source of energy. So it's about decomposition. Now let's return to tyrosine. Tyrosine is a non-essential amino acid. So it's mean they may synthesis in the human body. It's proteinogenic standard amino acid. For that reason, tyrosine need for synthesis of different kind of protein in the human body. For that reason, one of very important pathway utilization of tyrosine, synthesis proteins and peptides, for example, oligopeptides as here said. Next, tyrosine may involve in the specific metabolic processes coupled with synthesis catecholamines such as dopamine, norepinephrine, epinephrine, all that biological active substances, hormone epinephrine 2, synthesis from tyrosine, products metabolism of tyrosine. The specific product metabolism of tyrosine may be thyroid hormones like thyroxine and triiodtyronine, hormones of thyroid gland. And exists also one pathway utilization of tyrosine coupled with production of pigments in the human body, pigments like melanin. So this specific pathway utilization of tyrosine, but like all other amino acids, it may be used for synthesis protein, and undergo decomposition resulted from may be used for energy providing of cell. Energy providing of cell. Now, my dear colleagues, next question. You must know what metabolism phenylalanine and tyrosine, may be disorder at certain genetic disorders, may be undergo alteration during certain genetic disorder. Exists different kind of such genetic disorder, as albinism, as alkaptonuria, as phenylketonuria, tyrosinuria, and so forth, tyrosinuria. Let's pay attention for phenylketonuria. Phenylketonuria is one of most commonly genetic disorder coupled with utilization of phenylalanine. So, phenylketonuria. Next question, what we will say about here. Phenylketonuria. What's mean phenylketonuria? Phenylketonuria may be defined as diseases which accompanied by disordered metabolism of phenylalanine and its accumulation in the blood and in the urine. My dear colleagues, exist different causes arise phenylketonuria. And according to cause which bring to development phenylketonuria, exist some kind of phenylketonuria. I will say you only about two kind, but other clinical subjects we will say about more kind of the diseases. So let's return to phenylketonuria. Exists so-called classic phenylketonuria. Classic phenylketonuria is caused by a genetic defect of phenylalanine hydroxylase, so that enzyme don't produce, such as patient with such diseases don't possess expression genes of that enzymes, absent normal genes of that enzyme. This disease is inherited as autosomal recessive disease. And please note, heterozygotes appears normal. So heterozygotes individuals don't have any clinical manifestation of that disease. Exists also one kind of phenylketonuria. It's so-called atypical phenylketonuria. Atypical phenylketonuria is caused by defect of dehydrobiopterin reductase, which need for regeneration of a cofactor for hydroxylation of phenylalanine. See here, I again won't say you, that enzyme at atypical phenylketonuria present, but in order to that enzyme may provide its function, need tetrahydrobiopterin as that coenzyme. For reduction of dehydrobiopterin, need dehydrobiopterin reductase. But if that enzyme absent as a patient with atypical phenylketonuria, phenylalanine hydroxylase cannot provide process oxidation of phenylalanine into tyrosine, such as absent its normal needing for you coenzyme. It's so-called atypical phenylketonuria. Among that two kind of phenylketonuria, the most commonly classic phenylketonuria. For that reason, let's say about classic phenylketonuria. Please pay attention on that slide. So, a patient with phenylketonuria cannot be transformation phenylalanine into tyrosine, such as that enzyme absent, resulted from phenylalanine cannot involve in the biosynthesis of protein, cannot involve in the transformation into tyrosine. It cannot involve for that reason process breakdown to yield end product of catabolism, resulted from phenylalanine accumulate in the cell, accumulate in the blood, increase that elimination with urine for that reason, accumulate or increase that concentration in the urine too, And accumulation of phenylalanine bring to arise certain typical changes which bring to providing clinical manifestation of that diseases. My dear colleagues, what do you know, what you have to know about clinical manifestation of that diseases? Please note, the most typical clinical manifestation, mental retardation, which manifested as oligophrenia, pyruvate oligophrenia. If such patient don't undergo process treatment, usually the patient with phenylketonuria death till 10 years old. But if on time reveal the disease diagnosis, phenylketonuria, and begin treatment, such patient live as normal individuals. So about change metabolism at patient in the body, patient with phenylketonuria. First of all, accumulation of phenylalanine bring to arise acidosis, and accumulation of acidosis from other side bring to change rate of metabolic processes, such as for passing metabolic processes need certain pH, and decreased pH bring to decreased rate of different metabolic processes. Now, next, accumulation of phenylalanine bring to decreased energy supplement of cell, such as accumulation of phenylalanine bring to inhibition of glycolysis, resulted from decrease providing cell with ATP, and it's very important for nerve cell, such as function of brain, function of nerve cell require very good providing with ATP. The increasing accumulation of phenylalanine and its impossible transformation into tyrosine bring to decrease production of catecholamines. It's bring to disorder process transformation nerve impulse in central nerve system throughout appropriate synapses, catecholinergic synapses in central nerve system. And also accumulation of phenylalanine bring to disorder absorption other aromatic amino acid like tyrosine, like tryptophan by nerve cell, resulted from in nerve cell decrease synthesis different kind of neurotransmitter like norepinephrine, like dopamine, like serotonin, and other. All that changes bring to high, bring to high alteration processes coupled with function of synapses in central nervous system resulted from a rise mental retardation and oligophrenia. During process development of phenylketonuria, very important accumulation of phenylalanine. In such conditions, that concentration of phenylalanine sharply increased. Appear phenylalanine involved in unusual metabolic processes which not take place in the body of healthy individuals, healthy humans. Then concentration of phenylalanine at patient with phenylketonuria, phenylalanine involved in a transaminase reaction resulted from a rise phenylpyruvate. This reaction don't take place at normal concentration phenylalanine at healthy human. And accumulation of phenylpyruvate bring to its involved in the process reduction to yield phenyllactate and oxidation to yield phenylacetate. And phenylacetate later may involve in reaction conjugation with glutamine resulted from a rise phenylacetylglutamine. And please note, phenyllactate and phenylacetylglutamine at patient, in the body patient with phenylketonuria will be used as end product its catabolism and will eliminate from human body with urine. And it's very important to know, first of all, its appear in the blood bring to increase acidosis, such as all these products transformation of phenylalanine organic acids. And in second, appear that products such as phenylpyruvate, phenyllactate, phenylacetate in the urine may be used in diagnosis of that diseases. My dear colleagues, so I want to say you about diagnosis, laboratory diagnosis of phenylketonuria. Laboratory diagnosis of phenylketonuria may be coupled with screening revealing concentration of phenylpyruvate in the urine of patient. Usually that screening analysis uses at newborn child immediately after it born. In second, it may be revealing increasing concentration of phenylalanine in the blood and in the urine. And in third, it may be revealing defect gene which encoded enzyme phenylalanine hydroxylase via method PCR. That method is the most specific in the diagnosis of phenylketonuria. Next, about treatments, such principal treatment such patient. My dear colleagues, you must know what the disease phenylketonuria manifested immediately after birth such patient, and moreover, time beginning of the disease may be even during intrauterine development. So in conditions then fetus is not born at all. For that reason, in order to treat such patient, immediately early revealing the diseases, and immediately after revealing phenylketonuria, immediately you must prescribe such patient certain you must begin treat such patient. Then you undergo treatment newborn child. You must immediately stop breastfeeding, and prescribe such small patient special milk mixture, and that kind of milk mixture will contain milk what contain very small amount of phenylalanine. Amount of that phenylalanine need only for providing synthesis of proteins in the body such small patient. So, so then the patient with phenylketonuria will be gross and stop use milk as food. You must change diet such patient, and again diet such patient with phenylketonuria must contain a slow amount of phenylalanine, and such diet must use a patient with